Thank you to the organizer for giving me the opportunity of sharing with you our results on the importance of non-coding RNAs for muscle plasticity. As you know, skeletal muscle is an extremely heterogeneous organ because it is composed by different tissues, but also different myofibers types compose the skeletal muscle. Just to remember, myofibers are the smallest complete contractile system responsible for muscle metabolic and contractile traits. And we can identify different myofiber types. The slow contracting one with an oxidative metabolism. At the opposite, the fast contracting one with the glycolytic metabolism. And in the middle, the fast contracting with an oxidative metabolism. With the term plasticity, we intend that myofibers are able to move from one type to another using external stimuli. We can categorize myofibers according to the expression of myosin heavy chain, and within the parentheses, it is indicated the corresponding gene that codify for the specified protein. Taking in mind this extreme complexity of the skeletal muscle, the question is which type of results we can have using a bulk approach to dissect gene or protein expression. And the answer is that we have a result very similar to a smoothie with an homogeneous color without the ability of discriminating between each fruit that contributes for the taste or for the color. So several works were done to describe the importance of a single myofiber approach in the protein analysis, but all these works do not consider non-coding RNAs. I'd like to remember that this part of coding RNA is the most abundant part for the mammalia. The coding RNA is only the 3–4% of the transcribed RNA. The last part, more or less 78%, is non-coding. We can discriminate non-coding RNAs according to their length in small and long non-coding RNAs. Within the small RNAs, the most known RNAs are microRNAs. Previous speaker talked about. Just to remember, microRNAs are able to bind to their targets and are able to induce the degradation of their targets. Differently, long non-coding RNAs have several functions. For example, they are sponges for the microRNAs, or they are sponges for transcription factors that are able to avoid the binding to the mRNA, to the DNA. In this presentation, I talk about miR27A and 142, but we did also several work to dissect the effect of long non-coding RNAs in skeletal muscle atrophy. And if you want to to read something about, you can read our recent publication. It is known that there are several specifically expressed microRNA in skeletal muscle, like 133 and 206. Those are named myomers. And as you can see by this cartoon, it is also known that different microRNAs participate to the skeletal muscle development. So we started our study to identify RNA regulatory networks that can be involved in the metabolic plasticity of myofibers. It is already known that microRNAs participate to skeletal muscle phenotype, for example, targeting different transcription factors that can be involved in the phenotype specification. But taking in mind the first part of this presentation that described the complexity of skeletal muscle, we faced our problem isolating single myofibers from soleus and EDL of the mouse muscle. Each myofiber was classified according to the myosin heavy chain expression. This is a classification based on the protein expression. And since our question was to identify gene signature in single myofibers, each classified myofiber was used to profile the mRNA. In this slide, it is represented the first results we had. This is a heat map where in blue it is represented the low expression, and in red the high expression for each gene. The genes are represented in lines for each myofibers in the column. As you can see, different myofiber types have their specific metabolic gene signature. And for example, the transcriptional intermediate myofibers are enriched for genes involved in mitochondrial functions. As you can see, remembering that red indicates the high expression and the blue indicates the low expression, the intermediate myofibers express mitochondrial gene higher than glycolytic and oxidative myofibers. This is in accordance also with some publication from Stefano for that describe the protein expression. But remembering that microRNAs are able to regulate gene expression, we checked if the same myofibers have a specific miRNA expression. We identified 137 microRNAs, and as you can see, some microRNAs, for example, the myomer 1 and 133, are expressed in all myofibers, while other, like 206 or 208, are specific for the specific myofiber. At this time, we have the mRNA expression for for each myofiber. We provide 60 myofibers, and the microRNA expression for the same myofiber. We can integrate this information using bioinformatic approaches that are able to identify microRNA targets and associating the expression correlation, remembering that when a miRNA binds to its target, is able to degrade the target. So we expect that the correlation is negative between the expression of miRNAs and mRNA if the specific miRNA is a real target for the identified mRNA. We can construct a network that describes microRNAs and mRNAs interaction. And if we overimpose in this network the expression of microRNAs and mRNAs, we can, classify the function of the genes, we can see that the network is composed by two subnetworks, one describing the oxidative, essentially the oxidative metabolism, and the other describing essentially the glucose metabolism processes. In the same way, blue indicates the low expression and red the high expression. As you can see, the subnetwork that describes the oxidative metabolism is upregulated. It is most in red in slow myofibers, while the subnetwork that describes the glucose metabolism and processes associated with it is upregulated in fast myofibers. Obviously, in intermediate myofiber, we have an intermediate condition. I'd like to stress the fact that microRNAs, here represented by triangles, show an opposite expression with their targets here represented by squares. This is because the construction of the network and the analysis I described before. For the functional analysis, we selected specific microRNAs because their expression, because their, according to the targets, they should should regulate. And those are the microRNAs that we selected for the functional validations. With this slide, I'd like to stress the importance of single myofiber approach, because if you check the expression of the selected miRNAs in the soleus, that is the slow contracting muscle, and in ETL, that is a fast contracting muscle, you can see that all considered microRNAs are downregulated in the EDL, even if 142 is not significant. But if you check our results in the sequencing of microRNAs in single myofibers, you can see what you cannot see in the whole muscle approach. And here you see that the miR27A is more expressed in the intermediate myofibers, while the 142 is absent in the same myofibers. This information is not available using whole muscle. So, taking in mind that intermediate myofibers are the fibers more express genes involved in the mitochondrial function, we check that if the alteration of these microRNAs impact on the mitochondrial network. And as you can see from this slide, only the upregulation of the miR27A and 142 is able to increase the mitochondrial fragmentation. F index is the fragmentation index, and here you can see how here the mitochondrial are like a tubule, while here the number of spots is increased. Remembering the fact that the mitochondrial morphology is associated with the ability of mitochondrion to work, we checked if there is an alteration in the respiration after the upregulation of microRNA 27A and 142. We have seen that in the glucose medium there was the decrease in the maximal respiration only after the upregulation of miR27A. This difference is not statistically significant for the 142, while changing the medium for the cells using the palmitate to check only the mitochondrial respiration without the using of glucose, we showed a completely different results. With the upregulation of 27A, we have an increase of maximal respiration for the mitochondria, while with the upregulation of 142, we showed a decrease of the maximal respiration. After this result, we checked if after the modulation of the expression of these two miRNAs, we had a nutrient accumulations. And those are our results. After the upregulation of 27A, we had a glycogen accumulation, while after the upregulation of miR142, we had lipid accumulation inside the cells. Those are experiments in C2, C12 cells. The lipid accumulation after the upregulation of 142 was confirmed also with lipidomic analysis. After all these experiments in vitro, we did in vivo experiments inducing the upregulation of both microRNAs in skeletal muscle of mice. the PA staining of the muscles after the upregulation of each miRNA showed that only after the upregulation of microRNA 27A we had glycogen accumulation within the myofibers. And the most interesting result is with lipids, because after the upregulation of 142, we had an upregulation in the lipid content within the myofibers, but also after the upregulation of miR27A, we had a downregulation in the lipid content of the myofibers. So to dissect better the function of those two miRNAs, we decided to evaluate gene interaction after the upregulation of both. As you can see in this graph, where there is the list of genes that are downregulated after the upregulation of 27A or after the upregulation of 142, we selected specific enzymes for dissect the function of 27A, because I remember that after the upregulation of 27A, we had an accumulation in the glycogen within the cells. Those genes codify for enzymes involved in the glycogen breakthrough in the lysosome and in the cytosol. Regarding the miR142, we selected the most downregulated gene, the FNBC5, that is the precursor of irisin. Irisin is a myokine that is involved in brownization of adipose tissue. And I'd like to remember that this increases in energy expenditure. Remember that after the upregulation of 142 and therefore probably the downregulation of the irisin, we had a decrease in the maximal respiratory capacity of the mitochondria. We confirmed that the two microRNAs target PGM2, GAA, and FNBC5 also by protein checking in vitro, and also in vivo checking the expression of the corresponding mRNA in the skeletal muscles where microRNAs were upregulated. We confirmed also the interaction, the physical interaction between the microRNAs and their predictive targets using the luciferase assays that is based on reporter assay. But the last question we checked was regarding the results we obtained with the skeletal muscle and lipid content. I showed that after the upregulation of 27A, we had a decrease in the lipid content in myofibers, while after the upregulation of 142, we had an increase in the lipid content. We checked the expression of these microRNAs in skeletal muscles derived from high-fat diet mice. And as you can see, 27A is upregulated in all muscles.
5 minutes left.
Yes, while 142 is downregulated, but in EDL. So the mechanism we proposed is that in intermediate myofibers we have the upregulation of miR27A and the downregulation of miR142. The upregulation of 27A is able to induce fatty acid oxidation and therefore to decrease the lipid contents, and is able to block the glycolysis, inducing the accumulation of glycogen, while the downregulation of 142 avoid the blocking in these myofibers that are enriched for the mitochondrial function, the fatty acid oxidation. In conclusion, we collected all the genes expressed in myofibers. Here I talk you about mRNA and microRNAs, but we have also the collection regarding long coding RNAs, and demonstrated the importance of miR27A and 142 for the lipid utilization in the intermediate myofiber. I'd like to thank all the collaborators, people that have done this work, Francesco Chemello, Lisa Buson, and Enrico Alessio, and you for your attention. Thank you.
Thank you very much. Question, please.
I have a question.
Daniela?
Good morning.
Good morning.
Congratulations for this excellent presentation. I have a technical question. How did you induce the upregulation of this myomiRNA? Did you make a transfection, or did you of this myomiRNA, or did you use some compounds?
We did the transfection using liposomes in a manner very similar to the recent development of Moderna is developing anti-COVID vaccine in the same way. Also, we used the electroporation with the plasmids that are able to upregulate microRNAs, because with the transfection we are not able to follow the expression of the microRNAs, while using the plasmids we have a reporter gene that allow us to select myofibers that are expressing the plasmid.
Thank you.
You're welcome.